Hi everyone, it's Mr. Sinti, and today I'd like to talk to you about the different types of immunity. Now, if you're watching this and you're thinking, types of immunity, I thought there was only one kind of immunity. And the truth is, perhaps there is one kind of immunity that we have, but this particular video is going to discuss types, and so we're going to come up with some. And so, now actually, what we like to think about when we talk about immunity is the fact that, you get sick the first time, and then the second time around, following the exposure to the same antigen, you don't get sick, and that's what we mean by immunity. And sometimes that's called secondary immunity. When you get sick, and your lymphocytes need to build up and fight off the infection. That's called primary immunity. But actually, this particular video is going to look at different kinds of immunity, different types, and I believe there's four. So we'll take a look at that. So the reason that we have immunity in the first place is because of the fact that we have these memory cells, and so the memory cell is very important to the whole immune system because following this primary response to an infection, we have T and B cells remaining in our circulation. These can last for years. And so these memory cells do exactly what they're called. They remember, and their numbers are in high enough concentration that they're able to mount a substantial response, and so therefore you don't get sick the second time around. And so that's what that's all about. So secondary immunity means you don't get sick, and so that's the result. So the immune system's all about this, preparedness. And so it's as if the second time around, you're like, hey, I've seen that germ before, and so there's a quick response. And so the heart of the immune system comes from the fact that you have, for example, an antibody, and this is showing just the variable region. And so let me see if I can animate this a little bit. So this is the variable region on an antibody, and so this is, if you recall the structure of an antibody. If you don't, I'll refresh you. There's something called the heavy chain, and there's two heavy chains coming down like this, and they're held together by disulfide bridges like this. And then there's a light chain and a light chain like this. And so this particular region is the variable region, and then showing a close-up right here. Now this green area, let's see if I can go and pull out some green. This green area is the epitome of a particular antigen. So let's just draw some sort of bacterium like this. And so this is referred to as the foreign antigen right here, and it has this region called the epitome. And what's interesting about that is that these little yellow lines suggest that the variable region of an antibody is attaching via hydrogen bonds to the germ, and it can do a number of things. It could neutralize it, or it could make it a little bit easier for phagocytosis to occur. You could sort of attach to the epitome, and therefore, if this is a virus, make it unable to attach and insert itself into one of our cells. And so this is all a good thing. So it's all about antibodies. And so what happens is the primary immune responses, the first time you get sick, it takes several days for those B cells, which are secreting antibodies, to develop into effector cells and then plasma cells and then secrete antibodies. And so as you can see, in a couple of weeks, the concentration gets really high. But if the same, and that emphasize that, if the same antigen shows up again, notice how quickly the antibody concentration goes up, and therefore the secondary response is more rapid and more vigorous, and therefore you don't get sick. So that's what we mean by secondary immune response. There's a preparedness and an immunity, thus the name. And so what happens, like, let me go back for a second. Do you notice here that after the primary response, there's sort of a decline in antibody production? And so it's sort of, again, an analogy would be if your particular country was at war, and it takes maybe several months to get the numbers, the draft, and the numbers to increase and increase. But after your opponent has been defeated, you'd want to then suppress and sort of back it down. but you wouldn't want to totally take it all apart. You want to keep some bases around, for example, and that's what those memory cells are all about. But there is a particular type of T lymphocyte which is responsible to calming our immune response down, and this is called a suppressor T. It's an ordinary lymphocyte. It's a T lymphocyte, and it produces suppressor proteins. And the result of producing suppressor proteins is that it inhibits both the cytotoxic T and the plasma cells from reproducing. So it sort of causes them to calm down. And if the plasma cells start to decrease in numbers, then of course the level of antibody will go down as well. And so that's important, because you don't really want to maintain such high levels of concentration of antibody if the antigen is no longer present. It's not effective, and it's sort of a waste of energy producing proteins that are not necessary. So this is a classic example of a regulatory mechanism. So our immune system, you can't be naive and say, boy, I hope I have a strong immune system. Sometimes having a strong immune system is not producing a large amount of antibody when you don't want it. And so after the suppressor T produces those suppressor proteins, what happens to the concentration of the plasma antibody? It goes down, of course. And so this is what I mean when I get into a discussion of like different types of immunity. And so I'm going to be like sort of matter of fact about this and say this is the first type of immunity that I want to talk about. And it's something called naturally acquired immunity. Naturally acquired. And you're like, what is this? Naturally acquired immunity is everything that, if you've been watching previous videos that I've been discussing, in other words, and I'll review this if you haven't. In other words, naturally acquired active immunity is when you actually encounter a foreign germ like a bacteria or a virus and you get sick. And so there it is. The stimulus is the antigen. So this guy is the stimulus. And it's an active and it's naturally acquired immunity, meaning that as a result of getting sick, the secondary immune response is very strong. In other words, we have memory cells. They produce copious amounts of antibodies. And so this is what we'd call active. These are real memory cells. They're producing large amounts of antibody very rapidly. And so it's considered to be active. And so these cells are alive and they're producing, the memory cells are alive and they're producing a lot of antibody. And so the result of that is you don't get sick again. So it's a preparedness. So the second type of immunity is what I'm going to call artificially acquired immunity. So you're like, what's this artificial? Well, this is not getting the germ initially. So there's some germs out there that are particularly dangerous, and so we're aware of them, and so we have vaccinations against them. And so a vaccination, I'll get into this in a moment more detailed, vaccination is portions of the protein or weakened or dead germ inside a syringe, and if this is given to an individual, you're not going to get sick, but what it's going to do is when this dead pathogen comes in, it's going to allow your immune system, and I'll just summarize the whole process, it's going to allow the B cell to recognize this as being foreign and produce a tremendous amount of circulating antibodies, but more importantly, what it's going to do is it's going to cause these B cells to divide, and it's going to create a clonal army of memory B cells. And these memory B cells will circulate and be active through many years of an individual's life. And so it's artificial because you're not actually getting sick. The fact that it's called artificial is that you're getting a shot. But it's acquired, and so you acquire this immunity. So the stimulus is a vaccine, so it's a vaccination shot. And the result is that you have living memory cells that are able to respond very vigorously to a particular germ. So in other words, if you got a shot for chickenpox vaccination, you may not get chickenpox ever, even if you're exposed to it, because you have memory cells that are able to produce antibodies against that chickenpox, and so therefore you would have immunity. So a little bit more about vaccination. And so I mentioned a few things about it. And so a vaccination is basically causing your body to produce antibodies. Right here, it's antibody production. This is its goal, and the formation of memory cells without causing the disease. And so obviously you wouldn't want to give someone an injection of a particular disease if you thought that that was going to hurt them. So it's awesome. And so you're able to give a person a killed pathogen. So this is what this is. So this is a weakened or killed strain of a pathogen. And when this goes into the body, it causes the immune system to respond to that. And again, as I mentioned, it produces antibodies and memory cells, but it doesn't cause the disease. And so it's an absolute brilliant phenomenon. And so you're like, this is incredible. And so I mean, if I were to talk about how vaccines are produced, it's a totally interesting thing, and maybe I'll make a video about it. And so there's sort of an evolution about how we've gotten really good at creating vaccinations very rapidly, and it needs to be rapid in order to respond to the various germs that we feel are threatening us from year to year. Because the truth is, these flus and these germs will change, and they pose an ongoing threat constantly through mutation. And so I just wanted to point out one particular thing that I think is really cool, is that we're able to use recombinant DNA technology. In other words, we're able to take a gene from, and this is a segment of DNA, a gene from a particular germ, that you may know that the DNA of a gene is capable of producing a protein. So we wouldn't want to put the whole DNA in, but you can put a portion of this in to, let's say, a bacteria. And so we can use a bacteria and add a particular germ, add this particular DNA to the bacteria. And so what it'll do, it'll produce these particular proteins, and maybe these are the epitome of a particular virus, say for example, the viral coat. And so this is what we really want our immune system to respond to. And so this will make a tremendous amount of, say, viral proteins, viral epitome proteins. And then, of course, we will then purify this. And then this becomes what we use. These proteins is what we use in the vaccine in order to prepare the immune system. By injecting just those proteins, that's not going to cause a person to be sick, but it'll cause the immune system to respond to it. So it'll get the B cells to start cranking out antibodies and become memory cells. So how in the world did vaccination begin? And so this isn't kind of a lengthy, but I'll try to shorten it. It's kind of a fascinating story. This is a picture of a physician known as Edward Jenner. And so Edward Jenner was a physician who was working very vigorously, working on patients, very sadly, who were infected with the deadly virus of smallpox. And so you may know that smallpox was an absolute lethal killer, and it was highly contagious. And so basically, individuals developed these pusses and bumps on their skin, and it was contagious and it was lethal. And so these pox were really dangerous. And so there was smallpox, and it killed millions of people. And so, as it turns out, Dr. Jenner dealt with a lot of patients that had smallpox, and basically there's nothing you can do about it other than put them in isolation and sort of quarantine them away. And so now I may have the story incorrect, but this is what I've heard about this. And so interestingly enough, Dr. Jenner noticed that some of his patients came in who were milkmaids. And these milkmaids were women who were milking cows. And as it turns out, that cows developed a similar type of infection to smallpox. It was called cowpox. And as it turns out, that was contagious to people. Well, and it's like, well, who's going to catch cowpox? Well, individuals, women who were milking the cows. So as it turns out, Dr. Jenner noticed sort of serendipitously that that these women who were exposed to cowpox, and they had cowpox, although it wasn't lethal, when they were exposed to smallpox, they didn't get smallpox. And so they were immune to smallpox. And like, oh my God, that is a major, major observation, because being immune to smallpox means that you're going to survive. And so this is what humanity really needed. And so in order to sort of test his hypothesis that immunity was conveyed through the presence of smallpox, he literally went and he took some of the pus from the cowpox, and he deliberately infected a boy with smallpox. And I'm not sure if this was actually the case. This is what I've heard. So the boy came down with cowpox, but as a result of it, he didn't get smallpox when he was exposed to smallpox. And so this was the first vaccination. So why the word vaccination? Because it's Latin from the word vacca, meaning like cow. And so it's sort of an encowment. And so basically what he was doing was, if you give cowpox, which was similar in structure to smallpox, it allowed the immune system to develop B cells, and those B cells would then build in numbers. There would be enough antibody circulating in the person's body, so when the real killer came, smallpox, you weren't going to get sick from it. And so that's a huge thing, because for the most part, smallpox is no longer with us. The world was vaccinated against smallpox, and for the most part, it's never going to show up again. And so that's a huge success. And so the third type of immunity, if you're keeping track on this, third type of immunity is what I'm calling artificial, so that means a shot, artificially acquired passive immunity. So this is not active. You're like, wait a minute, what was active again? Active was when you actually had functioning B cells that live for years and produce a lot of antibodies. And you're like, well, what's going on? So sometimes a person lacks the time to develop immunity. And let me give you an example of this. Say you're walking on a hike, and you're like, wow, this is a great hike, and I'm loving it, and I'm climbing up the hill, and then snake comes and it bites you, and you're like, uh-oh, this toxic venom is going to kill me. But I'm sure after several weeks, my my immune system will respond to this, and I will recover. But unfortunately, you may not live more than a few hours after such a snake bite. And so here's another example. Say you're pregnant and you're exposed to measles, and this could be very harmful to your offspring. And so under these circumstances where there's not enough time to develop immunity, fortunately, we actually have antibodies that we could actually give a person. So this involves a shot. So this is why it's artificial. And what is it? It's not a traditional vaccine, but rather it's the actual protein, the immunoglobulins, that are going to be delivered against that particular snake venom or against measles. And so it's extremely rapid, and it's given, so these antibodies are given in the form of the shot, and so they're able to neutralize the measles or the venom so that you don't become dead from the snake bite. And so it's passive because it's not that you're developing immunity on your own. So it's considered to be passive, and it's artificial. And so the third and final type of immunity is going to be the fourth one, which is naturally acquired. And you're like, wait a minute, naturally acquired, but it's passive. What could this mean? And so it also involves a person getting antibodies, and that person usually is, you might have guessed it from the picture, a baby who's nursing. So you may know that the way the immune system is working is that it does take years to develop a very strong immune system, because in fact, what doesn't kill you makes you stronger. It's like, if you've heard that before. In other words, a newborn needs to become ill. Every child needs to get sick in order to develop that secondary immune response. But the truth is, some of these germs are pretty brutal. And so one way to strengthen a newborn's immunity is through breastfeeding. And like, how is that? Because what's in the milk is antibodies, in addition to the nutrients. Sorry about that. But in addition to the milk being a source of sugar and protein and liquid, there's very important proteins that are passed from the milk into the baby. And so this is what we call natural, because breastfeeding is natural. It's not a shot. And it's passive. And you're like, well, why is it passive? Because the truth is the stimulus is breastfeeding. And the result is that these antibodies are passed from the mother. And so the baby's getting all of these awesome antibodies that are circulating in the baby from the milk. And so the result is, though, that it's passive because the baby isn't really developing these proteins on their own. And so there's no effect on the lymphocytes. And so the lymphocytes aren't making their own. So the baby's getting sort of like the benefit of breastfeeding from the mother. And this particular type of immunoglobulin is usually immunoglobulin A. And it lasts for about a year. These are proteins. And so the baby needs to make her own proteins. And so sometimes you hear people say, and I've heard people say this, that if a person's not getting sick a lot, they'll say, wow, you have a very strong immune system. Were you breastfed as a child? And so, there's a little bit of truth in that, because I'm a very strong proponent for breastfeeding, because it does give a baby this acquired passive immunity. In other words, the baby is much less to be ill from breastfeeding. And so it's really important that a woman attempts to breastfeed. It's really, really important. But the truth is, it's not going to last a lifetime. And so it's really weird to say, to an adult that if they were breastfed, because these are proteins and they don't live forever. And so they're not really alive in the first place. And so really, it lasts for just about a year, and then you're good with it. Okay, so these are the 4 types of immunity in the human body. Okay, thanks for watching.